Okay, today we're going to do a UA. We're going to do just the strip analysis with the little chemical strips. So you see we have our box of test strips here. In lab, especially, it's important to make sure that these are the accurate strips and not expired demo strips. So these ones are within date. They're good to go. We're going to use them for a real UA strip test today. So one of the things you want to make sure after checking that the expiration date is good to go is that the container stays just as controlled as possible. So what that means is when you take the lid off, you're going to grab your one stick out and you're going to put the lid immediately right back on. And don't worry, it does tell you on the lid to replace the cap tightly and immediately after removing the strip. You don't want any air or moisture in the air or anything like that to get inside the test tubes. So when we do this strip test for the UA, what we're going to need is obviously the little test strip as well as some urine. So Sarah's provided some urine here from her dog Peanut that we're going to run a UA strip on. So before we actually get started with the strip analysis, it's important to be familiar with what it is you're actually testing. So I want to show you here, this is what the back of the bottle looks like. And each of these you can see are testing a different thing. So we have bilirubin, urobilinogen, ketones, proteins, checking for nitrate, glucose, what's the pH, any occult blood, what's the specific gravity, is there a presence of white blood cells? And then at the very bottom, our ascorbic acid test as well. And I know it's kind of hard to see. You know, it's kind of tricky. But each of these tests have a time limit that they have to be read. So for example, the nitrate, just the white box there, nitrate should be read at 30 to 60 seconds, and the pH down below also 30 to 60 seconds. But down here we get into the leukocytes, and that one can be read at 90 to 120 seconds. So again, it's just important to be familiar with your test and what it is you're testing, and then what is the time frame that you should be checking the strip against the result panel. So what I like to generally do, and there's a couple ways you can do this, if you have your urine sample just in a test tube, some people will just take their test strip, which you can see is nice and colored. Some people will just take the entire test strip and dip it down into the pee and then start a timer on when to read the test. What I like to generally do is pipette one drop onto each of the little pads. And usually what I'll do is I'll check what my time frame is. What's my one test on here that's going to run the longest? And I'll put the P on that one first. So in this one, it is going to be the leukocytes. So I'm going to add P at the very bottom, and then I'll go back and just do a drop on every other little pad. Okay, so really fairly simple test. You can, of course, do this over a trash can, over a sink, over a paper towel in the lab. That's fine. So this second to last one is my leukocyte. So I'm going to add my drop on there first. And put a trash can underneath just to make sure. So I have it on the leukocyte. Now I'm going to go back and add it to my other colors. Each little square gets a drop. And then I'm going to start the countdown, because the first ones that can be read, according to my bottle, is at 30 seconds. So between 30 and 60 seconds, the majority of these can be read. Okay, so just a little bit more time. Try to get that to look a little better for you. 10 more seconds and we can start. And what we're going to do to read this is we're looking at the colors and the test strip with the urine on it, And we're matching what color matches each of these results along each of these different tests. All right, so I think we can go ahead and start. We have our bilirubin, so we are just going to line this up and see how I have my strip lined up right next to our boxes here. So our bilirubin, it kind of looks like that first color. Our urobilinogen is the third one. It kind of matches that second color. And each of these colors have a little number. You can see those tiny numbers. And some of it say trace, some of them say negative. What you're going to do is determine what color lines up. So let's look at protein. So protein, my strip is light green, and the bottle that matches, or the bottle color that matches here, is also light green, and that says negative. So that's good. Glucose looks normal or negative. Then underneath that is my orangish one, the pH. It's about 6. And you just continue going down each of these until you have completed it. Now, it is important to record all of the stuff that you're determining. So we have, of course, our UA sheet that we are going to note some of these findings on. So again, going back to our test strip and the bottle that tells us the numbers or the score that we're going to write down, those are what's going to translate over here onto the UA form. So with his pH, we'll go back and double check that real quick. We don't want to wait too long. We said the pH was a 6. So in the pH area, we'll just write 6. Right? There was no occult blood. Bilirubin was normal. Our ketones, also negative. Glucose negative. We're just filling this in as we go. Okay. Now, of course, the second part of a urinalysis is to run a sediment. This was just the chemical analysis with the little test strips. And you should feel fairly confident in being able to run these.